All right, here we have a little biochemistry macromolecule review, and this sort of chart is found in your study guide for chapter 1, the very end of it. Remember that I have the type or category of macromolecule in my chart. Will be a little quiz to see if you can fill this out by memory. If I said 4 different categories using your memory of the nutrition label, I am hoping that you can come up with those 4. I'm going to try to have my chart semi in a straight line. Of course it's not. 1 is going to be the carbohydrate category. Carbohydrate. Second will be lipids, which we see as fats on the nutrition label. Third on that nutrition label will be proteins. And the fourth that's not on the nutrition label is going to be our nucleic acids. Nucleic acids. All right, hopefully you can read that. The next column I would like you to memorize for these 4 different categories will be the elements. So elements from the periodic table. Those are the elements I am speaking of. All right, so my chart is going to get large. And we're going to fill out different parts of the chart in a different order depending on what I want to write and where. Just kidding. So we're going to fill it out depending on the clue of whatever category we're looking at. So carbohydrate, that actually is a great way to remember what the elements in this molecule are. Carbo, carbon, hydrate, H2O. So this is a molecule that is going to be composed of carbon, hydrogen, and oxygen. The monomer unit or subunit is going to be a saccharide. Saccharide. And a lot of times you will hear of carbohydrates ending in -ose, but by no means is that the only way they end. So that's just one example, but that saccharide would be a subunit. An example of that could be a glucose molecule, and I suppose I'll add that to my category here. So we'll have glucose. And when I think of glucose, I typically think of a 6-carbon ring-like structure that has the O's and H's coming off of it. So we have O's and H glucose. I'm sure many of you will remember C6H12O6. All right. We will then look at what kind of cell structure is going to be made up of carbohydrates. So we'll have cell structure example. So I'm not going to ask for just a molecule that you'll find in the cell. Yes, glucose is going to be used as food, but I want to hear what structure is going to be actually composed of a carbohydrate, because there are plenty of them. Remember, one cell structure that we looked at that helps differentiate organisms based on kingdoms is going to be the cell wall. So cell wall, and I will just use cellulose, chitin, peptidoglycan as examples. Still there? Yes. Peptidoglycan. And you can also find carbohydrates in the cell membrane. So a lot of glycolipids, glycolipid, glycoprotein. These will be molecules that hang off the cell membrane. Lipopolysaccharide, next week when we get to bacteria. Lipopolysaccharide, you can hear that saccharide in that word. So that will round out that carbohydrate row right there. Let's move down to lipids. And lipids, when I think of lipids, usually I'll think of the monomer unit. And for me, the monomer unit is going to be a fatty acid. So fatty acids, when I think of a fatty acid, one of the fatty acids that is going to be most common is going to be that phospholipid. Remember, the phospholipid has that phosphate, PO4, head on it, and then it will have two fatty acid tails. So that kind of gives you an idea of what those fatty acids look like. A lot of them are carbon, 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 hydrogen, 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 oxygen, hydrogen, hydrogen. And if you have been wondering what the difference between a saturated fat compared to an unsaturated fat is, it happens to be in the amount of hydrogen that's present. If every single bond is filled up and bound to a possible hydrogen atom, we say that it is saturated with hydrogen. Saturated with hydrogen. However, if there are some bonds that are double bonds, a double bond has the electrons shared more so between those carbons, and now we don't have this hydrogen, then it will not be saturated, and that would be the difference, saturated with hydrogen or unsaturated. The elements then are the same as the carbohydrates, CH and O. However, a lot of times I will add on that phosphorus because the phospholipid is one of the most common types of lipids in our body. And cell membrane, you will find these, but all membranes, membranous organelles. So whenever you see that membranous organelle, like the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi apparatus, the mitochondria, those are composed of membranes that are that phospholipid bilayer, of course. And you already know the structure of that phospholipid bilayer with those embedded proteins as well as some possible carbohydrates coming off of the surface of those membranes as well. All right, moving on to proteins. When I ask students about proteins, probably the most common characteristic of proteins they can give me is proteins are composed of amino acids. So that tells you what the building block or monomer of a protein is. Amino acid. Then we are going to pull out some chemistry. When I think of an acid group, to me, I always think of COOH, a carboxyl group. With my amino group, I usually think of NH2. And then we will have an R group that's going to be found coming off of the carbon, actually. And you'll have your R group here that's going to differentiate between all of the different amino acids, 20 essential ones for humans. All right, using amino acid and these chemical groups here gives you an idea of what are the elements found in an amino acid. So let's keep it consistent. CH and O will be found. CHO. However, do you see any phosphorus? No. But now we do see some nitrogen showing up. And then I will share that in some amino acids, two primarily essential ones in humans, we have sulfur. So sulfur will be found in methionine and cysteine. There are a couple of different amino acids. Looking at an amino acid, I probably should have just had this structure over here. So I usually think of amino, my R group that changes, along with my COOH. Oh, you know what? Maybe I'm going to erase that. Sorry. And instead, I am going to cram some of the characteristics of proteins right into this box. I think I can do it. So with regard to structures of proteins, we do separate structures of proteins into primary structure, secondary structure, tertiary, and quaternary. Primary structure of proteins, sequence of amino acids. When you think of secondary structure, alpha helix, alpha helices, and beta sheets, kind of like I drew for that prion molecule. Tertiary is going to be that 3D. I always think that's kind of nice that it's the tertiary structure since it is three-dimensional. Sometimes we refer to that as globular. One of its most important shapes where it can work. And quaternary refers to multiple subunits. Multiple subunits. All right. I will hope to convince you that proteins are one of the most important molecules found in our cells. When we dehydrate cells, we find that our cells are actually composed of 50% proteins. That gives you an idea of the importance of those proteins. Well, I mentioned a dry cell, because dehydrated is too long to write. A dry cell has approximately 50% protein composing it, about 10% lipids, even though many people think lipids are quite common in the membranes, 10% carbohydrates, and then you have to start thinking about what does it take to make a protein. Well, when we think of the central dogma on producing these proteins, do you remember that in order to produce proteins, you have to start with the code found in DNA, transcribe that code into RNA, and then that RNA will be translated into a protein. So in order to make a lot of protein, does it make sense that you also need a lot of RNA? So we will have approximately 20% RNA and 3% DNA in our cells. And this is for the organic material in a dry cell. So moving on, where do we find proteins in cell structures? All over the place. We'll go ahead with embedded in cell membrane, all of those sodium-potassium pump, the ATP synthase machinery, the electron transport chain, all embedded in a cell membrane or in the mitochondrial membrane of eukaryotes. Do we also find them in cell walls? Well, if you remember peptidoglycan, peptido refers to the protein. So it is one of the components of peptidoglycan. And then proteins are also structural in nature, so those collagen strands, as well as actin microtubules, can have those form that cytoskeleton, the inner skeleton of a cell. All right, moving down. We're getting there. Nucleic acids. An example of those in the cell I already wrote up there, so let's just transpose that up here as well. RNA, DNA, and when I say DNA, we're thinking chromosomes. And another structure we'll learn about in bacteria will be plasmids. All right, thinking of the structure, since that's really what this is becoming in this column. The structure of RNA is really just typically a single strand. However, with that DNA, remember we think of a double-stranded helix. So there's your double-stranded helix of DNA making up that chromosome. The monomer subunit is a nucleotide. And a nucleotide is composed of three structures. One is going to be a sugar. Second will be a phosphate. And a third is going to be a nitrogenous base. All right, so the sugar is either going to be deoxyribose, or just the ribose. So it still has the OH group. So ribose or deoxyribose, depending on if you're talking about RNA or DNA. Phosphate will be the same for both, PO4 2-, 3- actually. And a nitrogenous base, remember your A, T, C, G, and U, nitrogenous bases. And we'll also find ATP is derived or has the structure of adenine in it. Finalizing this chart, finally, use what you know. Sugar, I do remember CHO in sugar, CHO. Phosphate, P. Nitrogenous base, N. There you go for your elements on that periodic table. So this is a great way to compare and contrast all of these macromolecules. What three elements do they all have in common? Carbon, hydrogen, and oxygen. The fact that they do have carbon and hydrogen will make them an organic molecule from a biologist's standpoint. So the hydrogen from a biologist kind of makes its way into that organic category because of one particular molecule that chemists will describe as organic. However, biologists think of carbon dioxide as inorganic, and that would be that CO2. Notice it is quite simple in structure, containing a carbon, but does not contain any hydrogen. All right, that's it.